Thank you so much. I really appreciate the invitation to be here. I understand that I'm in the way of a wine reception, so I will jump right in. My name is Reese Richardson. I am a recent PhD and now a postdoc at Northwestern University, studying the science of science, and I'm here on behalf of the collection of Open Science Integrity Guides, or COSIG. This is a project that we started that I think will be of a lot of interest to everyone here. And I'm going to go over what it is. Just for fun, I'm going to go over a few vignettes from COSIG, and then I'm going to talk about what's next for the project. So first of all, what are we working with here? The collection of Open Science Integrity Guides is sort of this open source textbook on performing post-publication peer review, or PPPR. You might also hear this called sleuthing, forensic meta-science. I like it calling it stewardship of the scientific literature. It's something that I believe we should all be doing. Is revisiting the scientific literature with a critical eye. Lots of people do this at scale. For instance, you already heard from Nick and James. But the idea is to look back at the literature and to leave peer reviews of papers, basically, that recontextualize findings and point out obvious problems, track down problematic papers, and try to create a record so that there's more information out there for people to make judgments when they're using these papers for future research or making determinations for hiring or what to invest in or what have you. So COSIG is a collection of as many tips and tricks that we can put in one place about how to do this thing. And it is fully open source. It's modeled off of an open source software project. So the idea is you can adapt COSIG however you like, and there is an open invitation to contribute to COSIG. So if you have tips that you think would be useful for other people that want to take part in post-publication peer review, we want you to contribute to this project, and it does not matter who you are. And to that end, COSIG is constantly expanding. So when we launched in June, there were 27 guides. Now we have 30. There are a couple of guides that are on the docket already, and the goal is for COSIG to keep expanding as long as it is useful. So these guides are fully versioned, and they can be distributed as one large PDF, or they can be distributed on their own. We want them to be used by research integrity officers, by educators, by working scientists, and anybody that wants to get involved in peer review and post-publication peer review. And as I said, they're distributed with PDF for maximum accessibility. You can find everything at cosig.net. And if you're remote, I encourage you to start looking there now. I think you'll find a lot of interesting stuff. But if cosig.net should ever go down, COSIG will live on as a open source software project. It'll be distributed as widely as possible. And like I said, the reason why we've made it that way is because we want it to be used. We want COSIG to follow the FAIR principles: findable, accessible, interoperable, reusable. We want them to be adapted. We want people to contribute. And we want anybody to do post-publication peer review. One of the reasons that we started this project is because when we talk about the work that we do as sleuths or forensic meta-scientists or people that study systematic fraud or what have you, a lot of people would say is, I think that what you're doing is fantastic. I think it's really valuable. I don't see how I could ever do it myself. You must have eagle eyes. You must have magical powers or something. You must spend hours upon hours at the computer just reading through papers. And you don't have to have any special skills to do this. Anybody, especially working scientists, are capable of doing post-publication peer review. And as I've already said, I believe that this is something that every scientist should be doing. but no matter who you are, you can take part in this. And you can even do post-publication peer review in disciplines you're not familiar with, so long as you have little tidbits of requisite knowledge so that you know what is acceptable and what is not. And the goal of COSIG is to distribute that knowledge. To that end, our guides cover basics of doing post-publication peer review. So like, how do I get started? Somebody here, although it's too early for me to know exactly what has taken place before my talk, but somebody here I'm sure has mentioned PubPeer. PubPeer is the eminent post-publication peer review platform where you can leave comments on any paper. We go over how you can leave those comments, how to write an effective PubPeer comment, etc., how you can report publication integrity issues to publishers, as Nick mentioned just a second ago. How can you respond to dismissive responses to integrity concerns? So things that make it super easy to get your feet wet. And then we have guides that will cover a number of disciplines. So I'll talk about a couple of these in a second, but guides on topics that it doesn't matter what discipline you're in, like plagiarism or image duplication or issues with citations. All of these things, people have already done a lot of work for how to detect these things, how to respond these things, etc. And those guides are all available. We also have subject-specific guides. So currently we have guides in material science, in biology, in mathematics, and statistics. And you can see a few of the examples of what we have here. And again, the reason that we make these guides open and available is because we want people to feel equipped to do post-publication peer review across a number of disciplines. So I'll go over a few tips and tricks today that will make it clear that like you don't need years upon years of experience and publications in material science to spot some very obvious problems in material science publications. Same thing with biology publications as well as mathematics publications. There are often issues that are glaring so long as you have just a tidbit of knowledge about that. So again, like anybody is capable of doing this. And we already have a large community of people that have contributed to COSIG. I myself am a maintainer along with my colleagues Yamar Ozturk and Salal Pirelli. And again, it is an open invitation to contribute. So if you have something that you think should be in COSIG, or if you had feedback on the guides, corrections to the guides, etc., feel free to reach out. You can leave an issue on our GitHub page. You'll find everything that you need to find at cosig.net. So again, like we really want people to use this, but we especially want people to contribute. We want this to be a project for the entire scientific community. So, what exactly is in COSIG? All this sounds great, but what are we actually looking at here? So I thought it might be fun to give a couple of vignettes for lessons that are included in COSIG that you might find useful just to prove how easy it is to get started doing post-publication peer review. So the first vignette is about EDX, or energy dispersive X-ray spectroscopy. This is a material science technique, and I'm going to venture a guess that most people in the audience are not material scientists. But basically, all you need to know about it is you bombard a sample with electrons, and then the elements in that sample will shoot off X-rays. And these X-rays are at well-defined characteristic energies. So this is an example of a EDX spectrum where you have energy of the X-rays coming off of it on the x-axis, and then you have the number of counts, how many X-rays at each of those energies have been recorded on the y-axis. So you can see that there are these peaks here, and these peaks are labeled with particular elements. So there's this peak at around 500 kilovolts, or 500 electron volts, excuse me, that is characteristic of oxygen, and that tells you that there's some amount of oxygen in the sample. Same thing with carbon, same thing with copper, same thing with tungsten. And importantly, elements will only ever produce peaks at these characteristic energies. So when you see an EDX spectrum, even if you're not a material scientist, you know that it's eminently fact checkable. And I'll go over how you can do that in a second. But let's put this into practice really quick. I'll teach you a couple of tricks that you can learn right away. This is a published EDX spectrum. If I were there in person, I would ask you to state if you can spot any issues with the spectrum right off the bat. But since I'm not there in person, I'll just let you appreciate the spectrum for a second. Realize that this would appear entirely plausible if you weren't looking that hard or weren't familiar with EDX, but there are issues with it that are immediately recognizable. First off, hydrogen will never, ever show up on an EDX spectrum. It is too light an element to actually produce characteristic X-rays. So right off the bat, if you see hydrogen on an EDX spectrum, something has gone terribly wrong. Also, carbon and oxygen will only ever have one peak. For carbon, that peak will be around 270 EV, and for oxygen, that peak will be around 500 EV. That peak will only ever occur there. So the fact that we're seeing carbon and oxygen peak, multiple carbon and oxygen peaks in the spectrum is a problem, but especially the fact that we're seeing them at 7,000 and 8,000 EV. That's just never going to happen. And finally, as I said, that carbon peak will only ever appear in that spot. So here they've labeled a carbon peak at 1,000 EV. It will only ever happen at 270. So there are multiple issues with the spectrum that are immediately identifiable now that you have that prerequisite knowledge. In the COSIG guide, we point you to some tables that you can use to reference where specific elements peaks should be. But the best way to do this is to download a software called DTSA2. And this is distributed by the National Institute of Standards and Technology in the United States. And it is a wonderful thing where you can actually plot out, here are the peaks that belong to each particular element. And you can immediately identify, like, this peak is not plausible. This peak should not be there in the first place, et cetera, et cetera. So I've commented on hundreds of papers that have issues with their EDX spectrum. And most of these, it is abundantly clear that they've just made it up. They've taken an EDX spectrum from somewhere else, and then they've labeled it with what particular elements they expected to see in their sample so that the reviewers, if there were any, would recognize that they did some sort of elemental abundance calculation. So on these, I usually present the spectrum and say, I see issues with this. One, there's hydrogen there. Hydrogen will never produce any peaks. Two, carbon and oxygen have multiple peaks, but will only ever produce one peak each, etc. And then I say, could the authors share the raw data for the spectrum and clarify how they assigned elements to peaks? One thing that we cover in our PubPeer commenting guide is that you should always try to make specific actionable requests from authors, because there are some issues that might be resolved by access to the raw data. And a lot of the time, they don't actually have the raw data because the publication is fabricated in its entirety. So this is a good way to keep the conversation about the content of the paper itself, and also draw attention to the fact that the data probably doesn't exist. So that's energy dispersive X-ray spectroscopy. And I hope that makes it clear how easy it is to do post-publication peer review, even if you aren't familiar with the discipline. This is a very complex topic that takes years upon years to actually master performing, but fact-checking it is really easy. And it's something that a lot of people can do. I myself am not a material scientist, but I wrote the majority of this guide and had it checked by Nicholas Ritchie, one of the developers of DTSA2, to make sure that the information there was reliable, and you can use this guide to spot issues in effectively any material science publication. So this one is a lot more general. This is the vertical line test, and you might already be familiar with this from elementary school maths, but the idea is, if you have data where you're recording something over time, or you're recording something where one value on the x-axis corresponds to one value of the y-axis, there are certain expectations for how that data should appear. So imagine that you're recording your height over time. Here's a mock-up of such a graph where you have time on the x-axis and you have height on the y-axis. This only makes sense if each value on the x-axis corresponds to a single value on the y-axis, because you can only ever be one height at one time. If the graph looked like this, where you have times on the x-axis that correspond to multiple values on the y-axis, that doesn't make a lot of sense. So this is not a mathematical function, putting this in mathematical terms. And the way that you can tell is that you can draw a vertical line through this trace that intersects the trace multiple times. So if such a vertical line exists that crosses over the trace multiple times, then the plot fails the vertical line test. And most line graph data is going to pass the vertical line test if it's legitimate. But if you're in a hurry and you're fabricating that data, you're hand drawing it, you're probably going to you may wind up with instances where the vertical line rule is clearly violated. So here's an example of that from a publication from this year in a journal about oral surgery. And this is a FTIR spectrum, or what they're claiming is an FTIR spectrum. And basically anything that gets called a spectrum, should be a mathematical function. Each value on the x-axis should correspond to a single transmittance or absorbance value on the y-axis. And here, that's clearly not the case. These peaks are probably hand-drawn, but in any case, they're completely unrealistic. So something went wrong here. Here's another example that's a bit less obvious. This is another FTIR spectrum, and you can see multiple instances where there appear to be these overhangs or backtracks where you could draw a vertical line through and it would intersect the plot multiple times. Here's another instance where the vertical line rule doesn't actually apply. So I said most data will obey this, but there are some exceptions, and you basically just have to use common sense to intuit when that's going to be the case. But here, these plots are actually tracking the movement of mice in 2D space. So the mice can move in any direction. So as a result, you would not expect these traces to obey the vertical line rule. So this would be an instance where the vertical line rule would not apply. So as I mentioned before, there are lots of different types of data that should obey the vertical line rule. Anything that gets called a spectrum, effectively anything where you would expect one value on the x-axis to correspond to one value on the y-axis. And you would be surprised how often this rule is violated. Because again, if we look back at these examples, it's not immediately obvious that the vertical line rule is violated. It looks like an FTIR spectrum, especially if you're unfamiliar with these things. But it's not something that you're going to zoom in on and see these things unless you're looking out for this. So the goal of COSIG is to make this more obvious, that these are things that you can look out for and that are eminently spottable. Okay. Next one is data duplication. So we've talked about image duplication. Image duplication is relatively easy to spot, especially now that we have lots of tools that make it easier to do automatically. I contend that that's because images are very data rich. And I think that when you have a lot more information contained in an article, it is easier to see problems with that information. Where there is little information contained within the data presented, then it becomes much more difficult. But there are still instances where you have line graphs and whatnot, where there is sufficient data to see that something went wrong. So, we'll talk about data duplication in terms of another material science technology. This is X-ray diffraction, or XRD. So the idea is you bombard a sample with X-rays at different incident angles, and different incident angles will return a different amount of X-rays corresponding to the spacings between atoms and whatnot. Again, it's really complicated to understand, but all you need to know for this demonstration is that no two X-ray diffraction patterns will ever be exactly identical. So here is a collection of 10 X-ray diffraction patterns that I got from my own university, Northwestern's X-ray diffraction core, where these are all the same sample taken on the same instrument, minutes apart from each other, under the same parameters. And yet, all 10 of these traces are unique. The noise in the spectrum is stochastic, it's random, and you're not going to see it repeated. It's a lot like fingerprints or snowflakes. They may look very similar, but no two will be exactly alike. And this is where we start to see problems, because where you would expect all X-ray diffraction patterns to be unique, you often don't actually see that. So for instance, here are two X-ray diffraction patterns, the black and the red one, and a standard that they're comparing it to. The standard just tells you here's where we expect to see the peak positions. These are supposed to be for two different materials. The black corresponds to an undoped sample. The 1% corresponds to a 1% doped sample. But if you look closely, you can see that these are, in fact, the same exact pattern. Not only are the peaks in the same position, but the noise in the background is exactly the same. This is entirely unrealistic, even if these were the same sample. If they're two different diffraction patterns, they are expected to be entirely different. So this is two diffraction patterns that appear, that are identical in the same publication. However, you can also see diffraction patterns that are the exact same and used across multiple publications. So this is a collection of 8 articles that I found earlier this year, where not only are they using the same diffraction pattern to correspond to multiple different materials in the same publication, but they're using this exact same diffraction pattern to correspond to dozens of different materials across a handful of different publications. So most image manipulation detection tools, as we covered in a different COSIG guide, will not pick up on this. But this is something that if you spend enough time looking at these patterns, you can spot these things. And where you see one of these duplications, you will usually find several. So these two examples have covered instances where the duplications are for the whole pattern. But let's say that you want to tweak the pattern a bit so that a peak appears in a certain position, or you want to remove a certain peak, or what have you. You might do something like what these authors did here. Again, if you saw this and if you were even peer reviewing this and assuming good faith on the part of the authors, you wouldn't necessarily get any alarm bells from this. But if you take a closer look at this pattern, you can see, as denoted by these red boxes here, there are regions of this pattern where the noise repeats. And there are probably 100 different data points in this small fraction of the pattern here. So it is incredibly unlikely that you would see this pattern repeating. And then we have a much more egregious thing here where there was probably some peaks that they wanted to remove from this part of the spectrum. So they just took the same a little blurb of noise and just repeated it 100 times in a row. And that is never, ever going to happen in a legitimate X-ray diffraction pattern. So when we come across these, again, the best response is to ask the authors, can you provide the raw data? And most of the time, they can't. But in any situation where you have high frequency noise, be it an electroencephalogram or an X-ray diffraction pattern or an EDX spectrum, you can spot these data duplications without really even interpreting what the spectrum means. And you can know that something went wrong here. And now this one's a little different. The last few that we've covered have to do with publications where clearly the data has been manipulated or something else went terribly wrong with how the data is presented. But these, there are also issues covered in COSIG that don't necessarily indicate manipulation or lying on the part of the authors, but nevertheless indicate some severe reproducibility issues that are worth bringing up during a post-publication peer review. So you may not be familiar with biomedicine, so I'll just cover this now. A lot of biomedical research is done using a reagent known as a cell line. And this is a population of cells that have been removed from a human or mouse or rats or what have you, and are kept alive separately from their original host and what's called a cell line stock. So most cell lines are immortalized. So they will continue to replicate and live forever under the right conditions, and they all originate from the same donor. So for instance, this is a osteosarcoma cell line called SOUS2 that is used very frequently for bone cancer research. There are hundreds of thousands of cell lines. There's this dictionary called Cellosaurus that maintains information on as many cell lines as possible. And you can see that they have 160,000 cell lines in their latest release. So there are lots of different reagents out there. The most popular cell line is actually the first cell line that was ever created. It's called HeLa. It is a cervical cancer cell line, and you might be familiar with it. It was obtained under unethical conditions, but that is largely beside the point. But if you've heard of HeLa, then you're already familiar with cell lines. And what's crucial about cell lines is that you make sure that you're working with the correct one. So this is a graph that shows hundreds of different cancer cell lines. So just for studying particular types of cancer, although cell lines don't necessarily have to represent cancers. And the cell lines are ranked according to how sensitive they are to a drug, a chemotherapy drug called paclitaxel. So on the y-axis, you have the amount of paclitaxel that it would take to kill half of the cell lines in a population. So for some cell lines, that is a very, very small concentration. And for other cell lines, even cell lines representing the same cancer, it takes 1,000 times as much to achieve the same effect. So regardless of what disease you're studying, of what organism you're studying, if you're not using a cell line that accurately represents the condition that you're studying, your results are probably not reproducible. So there's this entire thing called cell line verification where you actually make sure of the cell line that you're using actually represents what you want it for. Here's where that goes wrong. One instance is cell line contamination. I've spoken about HeLa before. HeLa is not only the first cell line and the most popular cell line, but probably also the most annoying because it tends to contaminate lots of other cell line stocks. So for instance, this is a cell line called BGC823. It is supposed to be a gastric cancer cell line, and it is very, very popular. But multiple publications have now shown that at some point this cell line became contaminated with HeLa. And no longer represents gastric cancer. And there are no known uncontaminated stocks of this cell line. So anytime you see this in a publication and it's supposed to represent a gastric cancer cell line, it is not actually doing that. There are plenty of other contaminated cell lines. If you want to read more about it, go to COSIG's guide on contaminated cell lines. But here's a few examples. Most of these, or a lot of contaminated cell lines, are gastric cancers. I'm not exactly sure why, but another example of a contaminated gastric cancer cell line, also contaminated by HeLa, is MGC803. Sometimes the cancer is, or the cell line is contaminated by another cell line that isn't even in the same genus, let alone the same species. So, for example, the B6 cell line is supposed to represent chicken cells, but was actually contaminated by pig cells at some point, and now is no longer a suitable model for chicken research. I've put these examples up not so that you should memorize them, but to make a point about cell line identifiers. You'll notice that they're basically just a jumble of numbers and letters. They mean something. So MGC 803 probably corresponds to the cancer center that collected it, as well as the number cell line that in this cancer center's stock. But they're largely meaningless. And this gives rise to another problem that is most certainly indicative of misconduct. Here's how we'll explore that. Here is a graph from a publication that came out in 2021 where they're describing the expression of this particular gene across a number of different cell lines. So across these 8 cell lines that they have here, there are 2 that are not known to be contaminated that probably accurately represent the condition that they're claimed to represent. And then there's another 3 here that have been contaminated, and in this case, they no longer represent gastric cancer cell lines. So here they're just assaying something completely different than they were assaying before. Then what of these remaining 3? Well, these cell lines probably do not exist. So these cell lines have names like BGC803, which you'll notice are very similar to the existing but contaminated cell lines, MGC803 and BGC823. What probably happened here was some paper mill saw that there were lots of publications that used these particular cell lines, MGC803 and BGC823, and wanted their publication to appear plausible. So they were writing down particular cell line names, and they made a typo where they typed BGC803, which is not a cell line that exists, but is an understandable typo and has happened previously before. But at some points, another paper mill reading those publications believed that that was a cell line that exists, and then used them to describe experiments independently of the existing cell lines BGC823 and MGC803. So these cell lines don't exist, or at least we haven't seen evidence that they exist at all. Usually there's a paper that tells you, here's how we established this cell line, here's where it came from. But they have a life of their own in the scientific literature. So last year, myself and my colleagues from the University of Sydney described 7 different cell lines that we have now seen collectively hundreds of publications claiming to use that don't actually exist. So you'll see, I mentioned BGC803 is probably the most popular non-verifiable cell line that we described. And this is probably just a mishmash of existing cell lines. And that seems to be how it happens. And sometimes these cell line identifiers do show up as just misspellings of one of the established cell lines. But a lot of the time, they're referenced alongside the established cell line as if they are their own independent cell line. And yet we know they probably don't exist. And that clues you in that not only is the publication irreproducible, it's probably entirely made up. And you'll notice that a lot of these identifiers are also mishmashes, are themselves mishmashes of contaminated cell lines. So there is a multitude of issues that you can spot with cell lines without really being familiar with cell biology in general. But if somebody's claiming that they're doing experiments for gastric cancer in a cell line that is known to be contaminated by cervical cancer, automatically that at least parts of this publication are not to be trusted. So this is a valuable thing that you can bring up in a post-publication peer review. And the thing that I like to ask the authors how they verified cell line identities. There are hundreds of services that will do what's called short tandem repeat profiling that it's standard practice to get this done on your cell line stocks so that what you're actually working with. And if anybody in the audience is a cell biologist, I recommend that you do this so that if you have contamination problems in your own stocks. But yeah, so that gives you an idea of all of the different ways that COSIG can be used. Just gives you a tasting of the different tips and tricks that you can find in COSIG. But there are now hundreds of pages of material in COSIG across dozens of topics. So I encourage you, take a look and see what you can find. I've certainly learned a lot in the time that we've been developing COSIG over the last year or so. So the goals for the future are to continue to expand COSIG's community. We want to bring more people into the practice of post-publication peer review. We want to arm experienced post-publication peer reviewers with new tools. We want to give valuable guides with domain-specific knowledge to editors and integrity officers. We hope that these get used for educational and training materials. We hope that developers use these to find automatic tools. We hope that librarians make this a part of their catalog. And we really want to, a big reason why we've made these accessible and put this out there is because we want more global representation in the post-publication peer review community. As we learned from Nick's presentation, a lot of the issues involving systematic fraud are seemingly concentrated on in locales where the scientific enterprise is not nearly as well funded as it is in like North America and Europe. And yet a lot of the attention on these issues is coming from North America and Europe. So we want people that are familiar with working in these environments to take part in post-publication peer review, both for the practice of post-publication peer review itself, and also because we think that that's what's best for a truly global scientific community. We also want to continue to expand COSIG, as I've hammered on already. If you have ideas for COSIG, if you have feedback for COSIG, please reach out. We would love to make you a part of the community. And we also want to continue improving these guides. These are not static objects, and certainly we do not cover everything that there is to know about X-ray diffraction and about cell line verification. So where there are points of improvement, there is room in COSIG. So yeah, thank you very much for listening. I really appreciate you giving your time today, and thank you for the invitation, and I'm happy to take any questions. Sam, you are muted though.
Thank you. Thank you so much. So that was a really, I wish this came sooner. This is such a great initiative, Reese, and I'm so glad to see it's actually making waves already. If I'd have known this 10 years ago, was going to be the future, it would have definitely got me out of the dumps a few times. So let's take a few questions from the floor. We have one here.
Hiya. So my question is, you say this is about a year old, 2 years old. I don't know how old this is, but has
This
it
actually
integrated?
launched in June. So it's less than a month old.
Oh, told you it's new. So does that mean, because I put this and then I put psychology and it doesn't seem to be as integrated just yet. So how long do I have to wait for this to be something that, one could use? Because it is a nice initiative. But I'm just curious because I typed psychology in Google and that, and it's not there yet. It's coming up, but it's not full.
Yeah, well, we would really love to include more guides on subjects like psychology. You saw the subject representation that we have now. We want this to be useful for the entire scientific community. So if you have ideas for issues that appear in psychology publication or tips that are useful when doing post-publication peer review on psychology publications, there is plenty of room in COSIG for that, and we would really value that contribution. So yeah.
Any other questions from the floor? I mean, it's interesting you say about psychology. That's usually the overrepresented content in this discussion about correcting bad science. It seems to be most of the ideas are coming from people in a background in the social sciences. I have a question, Reese.
Yeah.
One is so. This is, I mean, one of the issues which I sort of flagged with James earlier on in the day was that kind of what is the basic steps to flag issues that academics come across in reading papers. And you've mentioned a PubPeer and the sort of standard processes that they can go to. What can academics do to efficiently escalate issues if, say, they're not quite satisfied with doing a very thought out PubPeer comment or message or post or a blog post? Is there something in your guide, or how might you something in your guide that can give tips on how you can push for something to be corrected or for something to have wider awareness about?
We do have a guide on escalating issues to publication integrity teams at publishers. We are writing a guide currently about reporting publication integrity issues to institutional, so like university research institution integrity offices. Unfortunately, as Nick mentioned, there are not a lot of standards for actually processing this information and for handling complaints efficiently. And certainly when investigations do finish, you don't really know that that's the case because a lot don't actually observe a requirement that they report back to the person that reported something. A lot of doing post-publication peer review is frustration that little is being done. But it is, incredibly effective even to leave PubPeer comments, not only for the correction of the scientific literature. Something like 20% of all retractions that have ever taken place were preceded by a PubPeer comment. But also because, if you install the PubPeer extension, you can read through a scientific paper and see when it has which of its references have PubPeer comments, and it really enriches the way that you engage with the literature. I'm a big proponent of just putting more information out there, making more information accessible. That's a big part of COSIG and why we've pursued it. And just putting that information out there, like on PubPeer, like on a blog, is incredibly valuable by itself. So do not be discouraged by the fact that escalating to a higher place is often ineffectual. Um, but do be encouraged by the fact that more and more people are taking post-publication peer review seriously.
Any other questions? While I'm walking, I have one other question, and I know this is unfair because it's just come out, but have you got any case studies where people have used COSIG and it's led to a positive outcome, where they flagged something and something's been dealt with, you know?
I can tell you that I've now spoken with the integrity teams at multiple publishers who have made COSIG a part of their training materials for when they bring new people on. So hopefully that is good news that they'll be using these things for processing referrals, and hopefully expediting the process of handling retractions and whatnot. I've also seen a lot of PubPeer comments that seem to use language directly from the COSIG guides. And we've had a lot of people reach out and contribute material without being a part of the team that was originally developing COSIG. So we're seeing lots of positive signs that this is that the community is at least expanding. And I hope that we'll be able to see a concrete impact very soon, but it's looking very good.
That's amazing. I'm really pleased to hear that. Sorry, did you have yeah.
It's the same person speaking again with regards to the psychology. So do I use that email on the screen in terms of if I want to put ideas forward to push a psychology guide?
Yes,
behind
please.
that, because I really do want to bring some change, and I think the peer reviews, they're all just too complex, so maybe this might be an easier route to actually change quite a few things. So I'm going to use which email? The admin one.
You can email at admin@cosig.net will reach all of the maintainers. You can also email me at richardsonr43. You can reach us on the GitHub page. But yeah, admin@cosig is probably the best way to reach out. And if you don't hear back from us, feel free to pester me or anybody else. We're not hard to get a hold of, honestly. But I'll make sure that we follow up on that, because I do believe that it would be really valuable for more subject-specific guides. Some of the stuff that we cover in the general guides will also come about in psychology publications, but there's certainly a lot more ground to cover. So I'm really happy to hear that from you.
Fab. One more question. It could, I'll take this. This is the final one. But I'll remind everybody here that there is a free gap on Wednesday, I believe. So you could probably put your heads together and maybe draft an email to Reese to suggest some ideas if you want to use that time.
I was just wondering if there's any scope for potentially making some kind of digital tool for screening for some of the things you're talking about, because I think it's really amazing that there's information available to empower people to do their own checks. But I think also that leaves a bit of a gap where there are people that, for whatever reason, might not have the ability to actually do those checks themselves or retain that information. And also, as a mature student, I see a massive generational difference in approaches to things and the kind of utilizing and reliance upon technology. And actually having better technical tools available might make that more accessible rather than just information.
I totally agree. And that's one of the reasons that we've put out COSIG is because we know that there is a large developer community that want to make these tools available. There's certainly a lot of tools available for detecting image manipulation. And we have a whole guide on it. But we think that a lot of these ideas could be operationalized as automated checks. Certainly stuff like the data duplication, I also think that there is room for machine interpretation of common data formats like X-ray diffraction patterns and EDX spectrum and what have you. So yeah, I totally agree. I personally work towards developing a few different tools, and I hope to see way more of that in the future. And I hope that COSIG can be a valuable place to start for those.
Thank you so much, Reese. I think that about exhausts all the questions here. I don't believe there's any online, or maybe there is. I'll just make sure. I think everyone's just lavishing you with praise, so that's always a nice way to end. So, Reese, thank you so much. One last round of applause. I've just figured out, actually, I can have you all on the audience cam, I think. So if you can just see just the tops of people's heads there are more people down the bottom. If you just give him a wave. There you are. Thanks. Thank you so much, Reese.
Thank you so much.
Yeah, take care, and we'll see.